Today we're covering central dogma. We're looking at how um, DNA makes copies of itself, DNA information is converted to RNA, and RNA builds proteins. We'll also get into why these things are essential for life and have a little bit of practice on that transcription and translation process. The central dogma itself is a fundamental part of biology. This is something universal for all living organisms. Francis Crick, remember, he was the one that worked with Watson on the double helix model and some of that early DNA stuff. Proposed the central dogma first. To look at how genetic information flows like through cells, this DNA to RNA to protein process. So central dogma doesn't just explain that, it also gets into like how traits are determined for individuals, but also how your cells work and how they make the things they need. And along with that, how genetic information is passed down. We'll be covering inheritance soon, seeing how traits pass from like parent to offspring. And then it also helps explain how mutations can affect organisms, so how they function. Uh, Understanding central dogma in these processes also gives us a better understanding of how like genetic diseases work and how we can treat those diseases, as well as look at how organisms evolve over time. So we're going to look at this through something called the blueprint analogy. Um, is a good way to look at how how DNA gets handled in the cell. So think of DNA like blueprints for constructing a building. So those blueprints typically you're going to stay safe in the office. Stay safe somewhere off of the construction site, out of the job site, because we don't want those master instructions or blueprints getting damaged or lost. So they're going to stay safe in the office, or in this case, the nucleus. mRNA was going to be like a photocopy of those blueprints. So instead of sending those blueprints off to the job site where they could get lost, we're going to copy off just the parts of those blueprints that are needed for different jobs. And then the last part of it, protein, is going to be like the actual building. This is going to be what those blueprints tell them to make, what they end up constructing. So, quick review on DNA before we go through this process. Remember, DNA is deoxyribonucleic acid. It has that sugar or deoxyribose and phosphate backbone that makes up those sides of the ladder. And then the nucleotide bases, the adenine, thymine, guanine, cytosine, make up the rungs of the ladder. The adenine and thymine, apple in the tree, always pair together. The cytosine and guanine, car in the garage, always pair together. And those pairings are true just for DNA. So don't forget that part, because we will have a different combination when we talk about RNA. So our first process with central dogma is going to be DNA replication. This is what happens during interphase in the S stage or synthesis part. DNA gets unzipped. An enzyme called helicase breaks the hydrogen bonds in there between the two sides of the ladder and separates out those nucleotides. And then the cell is going to throw in or put in matching new nucleotides. So if the DNA strands had A, it's going to put a T with it. If it has a C, it's going to put a G with it to form two new complete DNA molecules. Each molecule is going to be half old, or we unzipped the original molecule with that helicase, unzipped it, and we've just added new nucleotides and completed them. These two new DNA molecules would be identical to each other. Skip that. Uh, our second process is transcription. It's going to look kind of like the replication because the DNA is going to split again, but instead of the entire DNA strand splitting, uh, only sections that are going to be used. This is where gene expression happens, where we're only using short sections or parts of that DNA. So that DNA is going to unzip, and then once it unzips, um, it'll start pairing up with nucleotides again to make an RNA strand. Now, the RNA is going to be a little bit different from the DNA because the RNA has a one different nucleotide or base than the DNA did. So RNA uses uracil instead of thymine. It will still pair with the adenine just like the thymine did, though. So the process will look something like this. These two blue ones are the original DNA strands. They're going to temporarily unzip here. That's what's happening in the middle where this RNA polymerase is. So they're going to unzip and we'll start getting RNA nucleotides matching up with the DNA. So you see those pairings. We have a C on the DNA, it pairs with the G on RNA, A with U, C with G, A with U. Even here on the DNA, we have the T, the thymine, it'll pair with an A on the RNA. Okay. There is a quick list of those base pairing rules over here, so you can go back and reference that later. When this happens, the cell is only using one strand of the DNA as a template for the RNA. And then this finished strand of RNA, once it's all built, is going to be called M or messenger RNA. There's a little more work that happens after transcription and that RNA is built from the DNA. A cap gets added to the start of it, and then the end of the mRNA gets something called a poly A tail. Someone was super clever when they named this. It's literally a bunch of A's added on the end. So poly, like many A's, so adenines. So the tail is literally many adenines added. There's a little other processing that goes into it. Things like introns are removed. That's parts of DNA and RNA that don't actually code for anything, so they're not needed for the next step. And then the exons, the parts that do actually code, are then uh, spliced back together. So the introns are removed, exons uh, move closer, get spliced together. So it shortens that mRNA a little bit. Once all that happens, the mRNA is processed, it'll leave the nucleus. So it's going to leave the office, and now that mRNA is going to the job site or the ribosome where construction will actually happen. Which takes us to the third process, which is translation. This will happen at the job site or in the ribosome, where we'll use that mRNA instruction to start building proteins. This step also introduces one more RNA. It's called tRNA or transfer RNA. And that transfer RNA is going to temporarily pair up with the mRNA uh, to line up amino acids to build the protein. So it'll be like this. This section down here, the AUG, UUU, CGA, is our mRNA strand. This is only a small section of it. And these parts coming in are the tRNA. And notice they're going to match up with the mRNA. A pairs with U pairs with A, G pairs with C. On a lot of diagrams for this, the tRNA uh, either looks like a key or it has kind of a T shape. Um, it'll it looks a little more unique than what this is. So you know that's the tRNA. The other thing going on here, these are amino acids, and they're getting carried in by the tRNA in a specific order. So they're going to line up based on the order of mRNA down here. As they line up, they're going to bond together and form a polypeptide chain. That's protein. So mRNA is going to be read in those groups of 3 nucleotides, and we call those groups codons, CODON, codons. There are 64 possible different codons, uh, but that's more codons than there are amino acids. So most of the amino acids we're looking at will have multiple codons that can make them. There are some other special codons, though, like this AUG that uh, tells the ribosome is the start of a polypeptide chain, and then we have some stop codons that tell it this is the end of that polypeptide chain. Remember that polypeptide chain, that's just literally a chain of amino acids that once they link up, they're going to start folding and they will form proteins. The specific proteins are going to depend on which amino acids and how they fold together, where they're being used. So misfolded proteins, again, this is like because we're getting wrong amino acids, can cause diseases. Cells rely on correct information. So when things don't get copied right or translated, transcribed correctly, or there's damaged DNA, it's going to result in the wrong amino acids and build the wrong proteins. So recap on it, we're going to go from DNA to RNA to protein. So that DNA has the genetic instructions for which proteins is going to build. We'll use that transcription to make an RNA copy of the DNA. And then translation will take that mRNA and start building protein from it. And the protein's important here, again, because all those cellular functions it affects, proteins work as enzymes. Those are organic catalysts that enable reactions to happen. This is how, like a lot of cellular functions, digestion, even that helicase that I mentioned earlier that unzips the DNA, that's an enzyme where is used to break those bonds. Proteins also get to be used for structure. They help move substances around in cells. Um, they receive signals. They help defense. Like there's tons and tons of purposes for proteins. So it's crucial that the right proteins get made. This is where that whole mutation thing is a problem. So mutations are just those changes in DNA, and it can be for different reasons. We have three types. That's substitution, where one of those bases, like the A, T, C, or G, gets replaced with another base. We have insertion mutations where an extra base, so like an extra A, T, C, or G, gets added in a DNA sequence, or a deletion where one of those bases is removed from a sequence. And it'll cause different problems. At best, we kind of hope for a silent mutation where, remember, there's more codons than amino acids. So hopefully it doesn't change which amino acid is made. Maybe it changes the codon, but the codon still makes the same amino acid. But it could cause a missense mutation where a different amino acid is made. And different amino acid means a different protein. Nonsense, where that amino or that polypeptide chain should keep going, but instead gets stopped early. Or frame shift, where because either a base is inserted or deleted, it changes all those three-letter codon groupings, so they're all now making different um, amino acids than they should be. So we're going to go through a little bit of practice with this, where we can start out with a DNA strand, something like this: TAC, GCA, ATG, GCT. And our first step is to transcribe it. We want to go from DNA to mRNA, so our messenger RNA. So our T would pair with A pairs with U, C pairs with G, G pairs with C, and so on. And we'll use a codon wheel to take those three-letter mRNA codons to determine which amino acids they'll make. I know this codon wheel kind of looks a little crazy. Don't let it intimidate you. It's not that bad, I promise. So our first mRNA codon was AUG. So we're going to start in the middle. These, the biggest ones here in the middle, that will always be our first letter. So we'll find our A, and then we're going to move out from the A to one of them that touch it in the second ring around here. So we have A, our second letter was U. Notice it still touches it, it's still in this section. So AU, and then our third letter in our codon was G. So AUG. Notice that G still touches it, it's still in this little section under U. So AUG gets us MET. That's going to be our start codon. Our second one, we have UUU. Same idea. We're going to start in the middle with this. Here's U. Go to our second ring. We have U again. And we're going to move to that third ring. U again. This is also a good example, though, of where if we had um, a silent mutation and maybe we end up with a different uh, base in here, maybe we would end up with UUC instead of UUU. They still will create or build the same amino acid, or GG, and then that third UCAG, all of those make the same amino acid. So those would all be silent mutations. We also have the possibility, though, where we could just one letter um, in any direction end up with a completely different amino acid. But that's our mRNA codon wheel. I think this is the easier way to find your amino acids. The other option is a codon table like this. Um, Same thing, you'll find your first base, your second base, and your third base, and cross reference across. This one is based off DNA instead of RNA. All right. So we'll stop there today. Um,